We're here to talk about a pretty heavy subject, which is ovarian cancer. And Amy's path through that disease, I have learned a lot about since then, is a pretty typical path. And that path was that she had no symptoms, that she discovered her ovarian cancer in the late stage, that she had this marathon surgery very quickly, and she experienced a brief period of remission. And then there was hospice. And that seems to be the path for many, many women that suffer this disease. And so I decided to form this foundation in memory of Amy and to concentrate on ovarian cancer. Because having seen the devastation of this disease firsthand, I wanted to change the outcomes for other women and other families. As most of you know, ovarian cancer is the first leading cause of gynecological cancer deaths and the fifth leading cause of cancer-related deaths in women. If ovarian cancer is discovered in the early stage, the chances of survival are roughly 90%. And if it is not, those statistics are pretty much flipped on its head. Our foundation focuses on early detection research because we know that early detection exponentially increases a woman's survival rate. So a little bit about the incredible work that we've done with all of your help. Since 2019, we have funded 5 ovarian cancer research grants with $350,000 in funding at 5 different research institutions. We have funded 4 separate ovarian cancer early detection research grants through the Foundation for Women's Cancer. And we are committed to fund an even larger, first of its kind, 2-year grant with the FWC for the upcoming research cycle. The Foundation for Women's Cancer is the charitable arm of the Society for Gynecological Oncology. Our grants are funding research selected by a prestigious scientific review committee to be the most promising early detection research. These grants provide young investigators the critical seed funding necessary to conduct promising research that can be leveraged to attract future grants from larger entities. And we've done that with Dr. Lengyel and Dr. Odunsi's help in really formulating the language necessary to attract the correct researchers for early detection. Significantly, as a group, the FWC's initial stimulus grant award recipients have parlayed the relatively small initial grants into multi-year, 6-figure awards, resulting in an average of a 23-fold return on the initial funding investment. And now, it took me a while to grasp what all that meant. Why are you supporting the Amy Krouse Rosenthal Foundation and not one of these huge, larger institutions? One of the reasons is we have a very small overhead. But what we're doing is we're issuing these grants in the amounts of $25,000 or $50,000. This year it will be a little bit more, which is small potatoes. A huge research, a peer-reviewed research study requires millions and millions and millions of dollars through major institutions. So we are giving young researchers the ability to get into a lab and do some work. And when they come out, they'll have hopefully a product to then give to or appeal to places like the National Cancer Institute, the Department of Defense, the CDC, the American Cancer Society, and other institutions to hopefully get those larger peer-reviewed studies going. So our work is really important, and hopefully we're going to move that needle as much as we can. Past FWC award recipients through the Amy Krouse Rosenthal Early Detection Grants include, and this is all only since 2020, Dr. Rebecca Porter at the Dana-Farber Cancer Institute, Dr. Sarah Corvino at the University of Texas MD Anderson, Dr. Mona Gao of the University of Southern California, and now our newest awardee, Dr. Sung from the University of Pittsburgh. And I won't profess to be able to explain to you all the details, nuances of their research, but I trust these 2 gentlemen over here, and they've done a wonderful job vetting these people for us. Additionally, Thanks to the incredible generosity of one of our donors, the 1111 Foundation, we are also funding our largest research grant at Dr. Lengyel's Ovarian Cancer Laboratory at the University of Chicago. Dr. Lengyel's lab is dedicated to improving our understanding of the biology of ovarian cancer metastasis and exploring the use of novel drugs for its treatment. The funded research study takes a basic science approach to identify a more effective and less invasive method to detect ovarian cancer masses. Findings from this study could lead to the creation of a liquid biopsy, which Dr. Lengyel will hopefully talk about a little bit later, a test conducted from a blood draw, which could become a simple yet powerful tool for diagnosing and managing masses, including aiding in the early detection, diagnosis of ovarian cancer. And like I said, I'm going to let our two distinguished guests tell you a little bit more about their research in a few minutes. Dr. Ernest Lengyel and Dr. Kunle Odunsi. Both doctors are recipients of numerous honors and awards for their work, including election to the National Academy of Medicine. The lists of their publications and their awards, NIH grants and appointments are so long and impressive that I'm just going to give you a short description of each of them real quick here. Dr. Lengyel is the Arthur L. and Lee G. Herbst Professor of Obstetrics and Gynecology and is the chair of the Department of Obstetrics and Gynecology at the University of Chicago. He is an expert in the diagnosis and treatment of ovarian cancer. His primary clinical focus is the surgical treatment of ovarian cancer, including advanced surgical techniques designed to improve patient outcomes. He runs a translational research laboratory at the University of Chicago Medicine to understand mechanisms of ovarian cancer seeding and is searching for novel blood biomarkers to differentiate a benign from a malignant ovarian mass to improve the detection of ovarian cancer. Dr. Kunle Odunsi is the AbbVie Foundation Distinguished Service Professor of Obstetrics and Gynecology, AbbVie Foundation Director, University of Chicago Medicine Comprehensive Cancer Center, and Dean for Oncology, Biological Sciences Division. Dr. Odunsi is a gynecologic oncologist who specializes in the treatment of ovarian cancer. As a nationally recognized expert in immunotherapy and vaccine therapy for cancer, Dr. Odunsi's research focuses on understanding the mechanisms of immune recognition and tolerance in ovarian cancer and translating these findings to immunotherapy clinical trials. Dr. Odunsi pioneered the development of antigen-specific vaccine therapy and next-generation adoptive T-cell immunotherapies to prolong remission rates in women with ovarian cancer. I am overwhelmed with gratitude for these two gentlemen, world-renowned physicians in this area that serve on our board, and we're really proud about that. So at this moment, I would like to introduce Dr. Lengyel and Dr. Odunsi.
Hello, welcome everybody. Thank you so much for inviting us, and thank you so much for inviting me. As some of you know, I was Amy's physician, and one of the things that really stayed with me, she had a disease course that was really rough, and she suffered from this terrible disease. But as I was sad about seeing her, that the cancers come back despite all the things that we are trying to do, she consoled me. Instead of me helping her, I remember she was in my clinic once, I don't know if you remember, Jason. And then she took my hand and said, "It's okay." I mean, all of you who don't remember, it's so Amy to do this and suddenly reverse and in her own way, the way she started and wrote books, she also challenged me and emailed me, "Hey, have you looked at this?" And I felt bad because she was ahead of me and found a study that was just published before I had even time to get to it. So I'm sad, but I'm also took a lot away from just going to know her, interact with her, and she had so good humor. And even if things were bad, she still knew somehow humor helped her, and I think that's what we can all learn from her, to get over challenges. And until the end, her brain was working really well, and she was a very sharp thinker. So that was also, she was an incredible partner for me as a physician. I just thought I wanted to tell you that. So I'm originally from Germany. I'm from Munich. I'm a gynecologic oncologist. So what we do is I'm trained as an obstetrician and gynecologist, and then we do 3 years of subspecialty training where we just learn how to treat women's cancer, which is mostly like ovarian, endometrial, vulva, and cervical cancer. And I have a research lab that focuses on 2 aspects. One is to understand metastasis. What Jason said, those ovarian cancers appear very, very late, and where already they have seeded to other part of the abdominal cavity. And we're trying to understand how cells that are normal in the body talk to cancer cells. And then we take from surgery, and we also collected Amy's tissues and Amy's DNA and put those cells together in the tissue culture dish and learn how they talk to each other and how they influence each other in a very dynamic system. And then use this system also to miniaturize it, because normally a tissue culture dish is about this size, but you can only test 1 drug at a time. So then we made it like, I don't know, some of you might have seen, they're like plates like this, where suddenly you can have 96 of those little wells, and you can test now 96 different drugs. And there are people that miniaturize it even more to 384 well plates, and suddenly you can use tissue from a patient to test many, many different drugs. And the cancer center that Dr. Odunsi leads, they have a core facility where we can do drug testing in a very automated way. And the second aspect of my research looks at how can we find ovarian cancer early. And since it's difficult, just think about the ovaries in a woman about this size. And it takes a lot of growth, so it has to go to this size to push in front on the bladder or push on the bowel in the back to cause any symptoms. And most women are tougher than we guys, Jason, I'm sorry. So they just ignore their symptoms. So, you know, often they come and they have a big tumor like this and they don't complain. I said, I always say, how can you do this, walk around and not even say something or complain? And the husband looks at me, I never knew that she had anything. So between kids and life and writing books and things, it's easy to ignore. And we're also scared. We are human beings, so we're scared of having. So all these things come together from different sides. And then you are also tough, and then, I'm just going to tough it out and not do. So that also contributes to the delay, where it takes about 9 months to have from the onset of symptoms to reaching a GYN oncologist. So what we're trying to do is we're using blood and have the hypothesis. So research, the way we do modern research is we don't just do and look for something. We have to have a hypothesis. This is perhaps a way to find ovarian cancer early. So our hypothesis is that in the blood, there must be markers that are secreted, like shed by this tumor, that can be detected then by methods to give us an early hint that something is happening. Now, I'm lucky to live in these times because, as you know, like, there's a lot of modern technology now. So technology that can detect 500, 800, 1,000 proteins in the blood at the same time, what we call proteomics. Or there's gene sequencing. Some of you know we have this big company in Chicago called Tempus that is able to sequence the blood of cancer patients, and suddenly you can detect 1,500 different genes and see if they are changed. But the problem is ovarian cancer is on nobody's mind. Most senators are guys, so they care about lung cancer, prostate cancer, and it's very, very little of the research funds goes towards women cancer. Dr. Odunsi just talked on our way here, like ovarian cancer, endometrial cancer, just really underfunded. And then if they fund women's cancer, they fund breast cancer. You see those buses and garbage cans with pink, and it's all over breast cancer because we almost cured breast cancer. 85% of all women who have breast cancer will not die from their breast cancer. While 85% of all women that are diagnosed with ovarian cancer will die from the cancer. So you see the difference. There's a highly deadly cancer, and there's very little funding for it. And then, like the people we train, they have no interest to go into a field like this because there's no funding. There's not a real good career path. Because if a university thinks they rather hire somebody who studies breast cancer or prostate cancer. So we're trying, in a very systematic way, every patient that walks into our obstetrics and gynecology department and has an adnexal mass, we take blood from these patients and their clinical information and ultrasound, and now use that modern technology that we didn't develop, that we apply to ask the question, can we detect that mass early? Can we say, is it benign or is it cancer? Because all of you would say, oh, I will give blood in a heartbeat, but doing a surgery or doing a big CT scan, that's a bigger decision, you need more time. So we're trying to make it very, very easy and lower the threshold that it takes for a woman to get a test. So we're trying to develop a test where we can diagnose ovarian cancer early. And actually, Dr. Odunsi and the Cancer Center has funded us what we call seed funds to look at changes in specific genes. There's something called methylation that's responsible for defining if a gene is highly expressed and then methylation is low, or if it's low expressed and then methylation is high. So that's a change in DNA that can be diagnosed and can be found and analyzed in a standard way. So we are applying this, and we've already collected blood from over 800 patients. And we are now slowly using this blood and this resource and all the clinical information from my clinical colleagues to compare women that did not develop ovarian cancer with women that had ovarian cancer. So we have two groups and are trying to see, is there a difference in this methylation pattern between those two groups that will help us to find this early. And I think that your association with the Foundation of Women's Cancer is really a very, very good one, because many smaller foundations like yours don't have the expertise to really, and it's so hard to give money, and with the pandemic and the inflation, you really want to put it to good use. But you have the advantage that they are like 8 or 9 experts that deal with these research questions all the time, that evaluate the people that you will give money for. And then they use this money that they get for ideas that are unusual, because if you have an unusual idea, the government, the NIH, will not give you money. So foundations like yours will allow to go after crazy ideas. In grants, they say they think out of the box, but some of those ideas are. But that's what you need, is you need really ideas. We can't just wait 20, 30, 40 more years. We need something now, and that requires a certain paradigm shift. The same way we saw it with most of you have an iPhone, I saw. Yeah, it was a genius idea to just have suddenly a phone where you can type and put your finger. So we need something like this. Most of you, like me, grew up without a phone going to school. You had a cable and were hiding to talk with your boy or girlfriend. So we need a paradigm shift, and support from you will allow for this to happen. We have the technology, we have the bright minds to do this, and we have the research question. So those three together with your support will make this happen during our lifetime. Thank you.
Yeah, thank you, Dr. Lengyel, for laying the foundation for what I'm going to talk about. So my name is Kunle Odunsi, director of the Cancer Center at the University of Chicago. So I came to University of Chicago about a little over a year ago, and from Roswell Park Comprehensive Cancer Center in another cold city, Buffalo, New York. But the main attraction for me really is the environment of the University of Chicago, ability to be able to work with individuals such as Dr. Lengyel and others, exceptional scientists and individuals. I think shortly after I came, I was contacted by, I think he called me to talk about this foundation. And without any hesitation, I did agree to participate and join the board. So I want to give you kudos. I want to say kudos to you for this important work, because ovarian cancer is an area that is in need of what you do. You are impacting lives, not only the present generation, but future generations to come. So for me, as director of the Cancer Center, I have broad oversight of the cancer mission, whether it's in clinical, education, or research for the University of Chicago. And what that means is that we need to pay attention to cancers like this, where there has been disproportionate funding, and make sure we're investing and keeping an eye on cancers like ovarian cancer. Moreover, I am biased because I'm a gynecologic oncologist as well, and I really want this field to advance, to have better outcomes for our patients. So I'm going to touch on 2 or 3 research areas from what we've done in the past and what we're going to continue to do here at the University of Chicago. So the first one is not really early detection. It's what I would call pre-vivorship. Pre-vivor, in other words, long before anything even happens. I'm sure we're all familiar with some genetic testing, BRCA gene, which is the main predisposition gene for ovarian cancer. So one of the things we did at Roswell Park Comprehensive Cancer Center in Chicago was to establish a familial ovarian cancer registry. It was established by one of my predecessors by the name of Stephen Piver way back in 1981. Dr. Piver had treated 2 sisters with ovarian cancer. And he asked the question, how can lightning strike twice? How come 2 sisters? And then I think this happened about twice or so. And that was when he started this registry way back in 1981. And this registry started to collect families where you have 2 or more affected first-degree relatives with ovarian cancer. And over the years, this registry has collected more than 3,000 families consisting of about 50,000 individuals. So when you collect the family, you get into full detailed pedigree information, grandparents, great-grandparents, what did they die of, and so on and so forth. And then around about 1989, the registry started collecting biospecimens, blood, saliva, any source of DNA. And in fact, by the time the BRCA gene was discovered, this registry contributed significantly to some of the biospecimens that were used to discover the BRCA1 gene, as well as BRCA2. Fast forward the last few years, where there has been tremendous impact from technologies. Dr. Lengyel spoke about genomic sequencing, where you can rapidly sequence the entire human genome. So one of the studies that we did recently, which actually was published very recently, a few weeks ago, is to ask the question from the registry for patients who have BRCA1 or BRCA2 mutations, are there modifier genes, are there additional genes that modify the risk? Because BRCA1 or BRCA2 mutation, the potential to develop ovarian or breast cancer is not 100%. The risk of ovarian cancer in a carrier of those mutations is really, for ovarian, it's really about 30, maybe 40%, lifetime risk, not 100%. So what modifies the risk? So we were able to do something called whole genome sequencing because of access to this unique set of biospecimens from families where you have 2 or more affected first-degree relatives and where you have clear evidence of BRCA mutation. What are the other genes that are modifying the risk? Of course, we know there are some other genes, but this type of registry allows us to uncover and detect and discover new genes that could modify the risk. So what we found was another new gene that actually leads to an earlier age of onset. So I'm not going to, quiz you about the name of the gene, but it's called PPAR gamma. It's a gene that modifies the risk. If you're a BRCA mutation carrier, the age of onset significantly reduces to about 48 years, whereby those patients develop ovarian cancer earlier, whereas the standard for BRCA1 carriers is generally around the age of 55 years or so. So you have earlier onset, about 7 years lead time, if you carry both BRCA and the PPAR gamma gene abnormality. Okay? We think this is just the tip of the iceberg. There are probably other modifier genes that we need to still discover. So we're now in the process of trying to establish that similar registry here in Chicago, at the University of Chicago, where we can further build on the registry in Buffalo, New York, as a collaboration between the 2 institutions, so that we can expand the registry base. Moreover, the registry in Buffalo, when we look at the demographics, it's about 99.5% Caucasian. Speaking to this issue of healthcare disparities. So we think we have a unique opportunity in Chicago to be able to enroll into that registry and obtain genetic information of modifiers of BRCA mutations in more diverse populations based on our geographic location in Chicago. So that is an ongoing exercise. We're in the process of putting together the IRB protocol and get all the agreements in place in order to be able to establish this registry. We think it's a powerful mechanism for detecting novel genes, and it's also possible that these new genes could allow us to develop new therapies as well by targeting some of these genes. So that is, so what I just described is kind of a side project. My main interest is really in immunotherapy. And several years ago, we asked the question, can the immune system recognize cancer? Can the immune system recognize ovarian cancer? Why is that an important question? If you think about it, cancer cells are part of self. So the immune system normally will not recognize self because that's not how our body is trained. The immune system, when it does that, then you have abnormality. You have autoimmune disease. You have multiple sclerosis. You have all kinds of autoimmune disease. The immune system will rather die than try to recognize self. So there are so many built-in safety mechanisms to prevent the immune system from suddenly attacking, let's say your brain or your heart or your so many fail-safe systems put in place. A lot of them. These are called mechanisms of tolerance. The immune system has to tolerate self. So cancer cells are part of self. So the question becomes, how do you break tolerance? How do you, first of all, show that the immune system can recognize self? So a number of very elegant experiments conducted by some of our colleagues way back showed very clearly that, in fact, there's a state where the immune system is constantly surveying the body for abnormal cells. Immune surveillance. So that every one of us sitting in this room probably has some abnormal cells, but the immune system is able to keep them in check. Then there comes a stage where the cells are growing and the immune system is trying to recognize them, but immune system is beginning to fail to recognize them. And then there comes this state of equilibrium where everybody is coexisting, the cancer cells and the immune system, everybody is happy, everybody is going their own way, it's not causing any problem. The final step is when you have immune escape, the cancer cells escape from immune attack. And that's when you now have gross tumor cells. You have cancer that has spread all over. So when we see a patient with ovarian cancer, as we previously mentioned, it has spread. It has escaped from immune attack. This is not the state of surveillance or equilibrium. This is a state of escape. But can you still use the immune system to do that? So we embarked on a series of studies whereby we were trying to identify that even at that stage, are there targets of immune recognition? Are there things that the immune system can see on a cancer cell that is present on the cancer, not on normal tissue, that you can then use to trigger the immune system to recognize? Because normally the immune system readily will recognize pathogens, virus, COVID, or any other virus. So as a result of those studies, we were able to develop vaccines to try and trigger the immune system, especially in patients who are in remission from ovarian cancer, to say, can we minimize the risk of relapse? Or if relapse is going to occur at all, can we prolong it at least? Can we delay the relapse? So we conducted a number of studies over the years. The studies did not get to randomized trials, but based on all of the data, we show that, yes, we can generate strong immune responses against ovarian cancer. And number two, we can potentially prolong remission in many of these patients. But where I'm taking this story is the current state, which is ovarian cancers, based on all of the clinical trials. If you are following all of the studies that have been done in ovarian cancer, so far has not been very responsive to some of the current immunotherapies. Immunotherapies, I'm sure you see Keytruda on television all the time. There's a drug called Keytruda. There's a lot of new drugs these are immunotherapies. They've been very effective for many cancer types, such as lungs, melanoma, kidney, bladder, name it, breast, not effective in ovarian cancer. So we think that ovarian cancer is going to need a big, you're going to need it's almost like firing a big gun, in order to be able to attack ovarian cancer. And what is this big thing? You need large numbers of immune cells to get into the tumor and be able to survive and stay fit in the tumor environment and kill the cancer cells. So these are called T cells. Okay, so we need large numbers of these T cells to efficiently get into the tumor and be able to persist, because once they get into the tumor begins to fight back and begins to throw stuff at them. So one of the things we've embarked on is to I'm sure you've heard about CAR T cells. Here, we're not talking about CAR T cells. We're talking about identifying specific cells where the T cells, making cells where the T cells can actually recognize an ovarian cancer cell and destroy them. So what we've done is to clone out specific T cells that can actually be able to recognize ovarian cancer and use that information to engineer additional cells, okay? So that now we can take cells from a patient, manipulate it, generate billions of T cells that can recognize ovarian cancer, okay? So this is called cell therapy. This is what we think ovarian cancer is going to need as one of the strategies, whereby you need this large number of cells to be delivered into ovarian cancer. There are a lot of obstacles. So we're testing different approaches, different ways of engineering the cells, different ways of making sure the cells are protected once they get into the tumor, so that whatever the tumor throws at them, they can survive. So many angles to this story, but we think that's going to be a key element in a few years from now, if we're able to crack that code. We think ovarian cancer ultimately will be able to deploy immune cells to attack, but there's ongoing work. There's work to be done, and that's some of the type of work that my laboratory is doing. Finally, I have to say, I did not have the privilege to meet Amy like Dr. Lengyel did, but even on our way, we went through Wikipedia. We were reading and looking at what was written about her. It's just amazing. I did not have that privilege, but again, thank you for what you are doing, for your support. It's truly making a difference, well beyond what you can imagine. So thank you.
Yeah, so the question was why CA-125, which all of you might have heard is a tumor marker. CA-125 is very good for advanced cancer, but it recognizes only 45 to 50% of early cancer. And it's also unspecific in women that have endometriosis, or even in COVID, we saw that CA-125, every time you have an inflammation, you get an elevation of a CA-125 because that's an acute phase marker. So actually, you harm women if you test CA-125 because they undergo a lot of unnecessary surgery, which has its own risk factors.
Thank you.
Yeah.
I have a question about collaboration. You were talking about how you were going to expand the data. And it sounds like there's obviously some bureaucracy that's going through that and everything. how is it that we can't multiply on that? How is it that we can't work with other hospitals and really come up with the numbers that we need to really look at this and get a data set that's easier to work with?
Yeah, that's a good question, and that is something that we always try and navigate because of a lot of federal rules. There are rules surrounding data sharing, there are rules surrounding HIPAA, there are so many of these things that we need to navigate. But fortunately, the one that I described between Roswell Park and University of Chicago, these are two NCI comprehensive cancer centers. So we have ability to collaborate a lot easier because, so for example, the IRB protocol that I described, the Roswell Park Cancer Center we're going to maintain the IRB from Roswell Park, the Institutional Review Board, will be maintained from Roswell Park, and that makes it a lot easier for us. And then we'll combine, right, so the same protocol for intaking those families where you have 2 or more first-degree affected relatives. We're using exactly the same protocol, putting information in the same data system. So I think it's going to be a seamless collaboration, which I'm hoping will finalize very, very soon. But you are absolutely right. If this was going to be between Chicago and Northwestern, we'll have to go through a lot more hoops. Actually, maybe Northwestern, Rush, because Northwestern is an NCI center. If this was between Chicago and, let's say, Rush, we'll have to go through a lot more hoops before we can get this done. Yeah.
Are there other cancers that don't respond well with immunotherapy, that you need this super cell thing besides ovarian, that you could partner with these other cancers that are also needing that type of supercharged cells?
Yes, so good question. So the couple of them, actually, one is pancreatic cancer. The problem with pancreatic cancer is it's almost like there's a shield on the cancer that even prevents, so even if you deploy large numbers of cells, they can get in. It's almost like there's a wall. If you look under the microscope and look at a pancreas cancer, you can see immune cells come to the edge, but they are not able to get in. So we need to do a lot of things to be able to break that wall so that the immune cells can get in. So that's one example. Another one that hasn't been as successful actually is prostate cancer as well, where immunotherapies have not been as successful as some of the other cancers that I mentioned. So at Chicago, we have what we call a cell therapy program, where we're looking at, and in partnership with the Pritzker School of Molecular Engineering. These are molecular engineers looking at different strategies to build the super T cell. Again, some of the cells will depend on the type of cancer, depend on what is the predominant thing you want to achieve in this type of cancer versus the other one. If I was to design a cell for pancreas, I would design something that will not only arrive at the tumor site, but also break down the wall to be able to get in. So, so we have molecular engineers looking into some of these issues.
What comes to mind immediately after that is, I am just so glad that there are people in this world that are a heck of a lot smarter than me and so devoted to their work. It really is just incredibly, incredibly impressive what people do each day, when you learn about them. So Thank you both so, so much for your words, and mostly for agreeing to be on this board. It really means the world to us. Thank you.